cool. Pei, you probably wanted to introduce a little bit about yourself.
Okay. Could everybody hear me?
Yeah.
Okay. Hi everyone. My name is Pedro. I'm a exchange graduate student in Pinwen's lab. So the topic I want to share with you guys today is the preoptic anterior hypothalamus estrogenic regulation of body temperature and energy homeostasis. So here are some backgrounds. To evaluate the estrogenic effects on the body temperature, the most common method is to remove most of the endogenous estrogen by gonadectomy or ovaryectomy. And as shown in the left upper figures, the estrogen cycle, as the estrogen cycle going, the estrogen level is dynamic changing. In the proestrous phase, the level is the highest. So combined with the body temperature and the physical activity monitoring, which have been shown in the left bottom figure. This research compared the GNDS mice and the control mice in an entire estrous cycle. As we can see, compared with the control group, the GNDS females have the lower body temperature and the lower physical activities in the whole estrous cycles. Wait, sorry. Sorry, I probably put the wrong PPT. 1 second. Sorry about that. So as the left bottom picture shown, this research compared the GNDS mice and the control mice in an entire estrous cycles. As we can see, compared with the control group, except the meta-estrous phase, the mice body temperature and physical activities are significantly lower in the dark cycle, while they are higher in the light cycle for the proestrous cycle, which indicates that the mice lost the ability to regulate the body temperature. And interestingly, as the right pictures show, other research subcutaneous implant the estrous pellets to the OVS female and monitor the body temperature in different environmental temperature. And they find that in the cold temperature, the estrogen increased the mice body temperature and it decreased the body temperature in the hot temperature. So this data suppose that the estrogen is essential for maintaining and regulating the body temperature. Then let me briefly introduce the brain region we focus in this study. So in the central nervous system, the region regulates the temperature, food intake, parenting, and the thirst is the area called the preoptic anterior hypothalamus, which is also named the POAH. So the POAH include the medial and lateral part of the preoptic nucleus and the anterior hypothalamus and the nearby region of the septum. POAH is responsible for the thermal regulation and receive the nervous stimulation. Firstly, we are wondering what's the effect of the directly infused estrogen in the POAH. As shown in the lab picture, the POAH site-specific infusion decreased the 4 hours and 24 hours food intake, What's more, our data showed that the estrogen could also decrease the body temperature after the infusion. So estrogen acts on binding with its two subtypes of receptor, including the ERα and ERβ. They have a high similar structure. Both of them contain N-terminal DNA binding domain and a C-terminal ligand binding domain. Binding to cognate ligand cause a change in the receptor, cause the difference in their binding region and function. As shown in the picture, showing the picture, there is only 18% of the homology between them in the AF1 and AF2, which initiate our shot to investigate the role of the ERα and ERβ signaling in the POH. So firstly, we performed the IHC and the in situ hybridization, finding a very high level of the ERα and the ERβ in the POH. Moreover, we stained the ERα in the ERβ GFP mice to find out their expression pattern. And as shown in the pictures, we find that the ERα and ERβ could express in the POH neurons. To count, actually over the 92.5% of the ERβ neuron could express the ERα. This data make an interesting question. We want to know how they respond to the estrogen and how they coordinate or compete. Therefore, we perform a PET scan to monitor the response of the ERβ neuron after the estrogen treatment. So as shown in the figure, in this ex vivo experiments, we find that the estrogen cause a mixed action in the ERβ neurons. Specifically, we find 21% of the neuron didn't respond to the E2, while 42% of the neuron are inhibited and 36.8% of the neuron are activated. And this data indicates that those two estrogen receptors may cause an effect in a different direction. To prove our hypothesis, we further treat the ERβ neuron with the ERβ or ERα agonist DPN or the ERα agonist PPT to further confirm the karyotypes of these neurons. And as shown in this picture, we find that the ERα agonist activate, but the ERβ agonist inhibit the ERβ POH neuron, showing a competition on mediating the neuron activities. And since we find that most of the ERβ could express with the ERα, we are wondering whether the estrogen inhibit the ERβ neurons through the ERβ. So we firstly block the ERα by using the BHPI, which is ERα receptor inhibitor. And we find that this time the estrogen caused all of the neuron inhibition. Well, the estrogen failed to affect the neuron if we block both the ERα and ERβ. Therefore, the estrogen inhibits the ERβ POH neuron in an ERβ dependent manner. And since these two receptors showing a strong and independent effects, We are wondering if the ERα and ERβ counterbalance between the ERα and ERβ have the physical relevance in different conditions. So by testing the membrane potential, we find that after 2 weeks of high-fat diet feeding, the ERβ neuron resting membrane's potential significantly increased, which means the neuron stability is lower. And we also find that although there is no difference for the ERα or ERβ between the chow or high-fat diet feeding, the ratio of ERα to ERβ is significantly lower after the high-fat diet feeding, showing an increasing tendency of the ERβ. Then similarity, we treat the high-fat diet feeding ERβ GFP brain section with the estrogen, and as you can see here, the estrogen caused either no difference or inhibition of the ERβ neuron without the activation. And if we compare with the child diet feeding data, we find that the energy reserve increasing. With the energy reserve increasing, the estrogen cause a major effects of the neuron inhibition. And similarity, we are interested in the estrogen effects on the ERβ neuron and different temperature exposure. And firstly, we find that compared with the room temperature, the cold exposure decreased the neuron stability, while the thermal neutrality increased the neuron stability. Then we treat the brain section post 2 hours of the cold exposure with the estrogen. And we find that most of the neurons were inhibited. Well, as we can see this picture, most of the neurons are activated by the estrogen and the thermal neutrality. And as summary in this picture, as the ambient temperature increasing, the temperature tend to activate the ERβ neurons. So based on the preliminary data we got, we hypothesized that estrogen acts through the ERα and ERβ in the POH to modulate the energy homeostasis and the metabolic adaptation to the environmental challenges. And according to our hypothesis, we have 3 aims to go. So firstly, we are aimed to determine whether the estrogen drives the temperature-dependent energy homeostasis by activating the ERα POH and inhibiting the ERβ POH neuron. So for this purpose, we generate a mouse model which allows us to selectively activate or inhibit the ERα neuron or ERβ neuron in the POH. Specifically, we use the ERα or ERβ Cree as a mouse model. So for the selective activation model, we stereotactic and bilateral inject the Cree-dependent virus, the AAV DIO-HN3DQ-M cherry to the POH. The HN3DQ is a modified form of the human M3 muscarinic receptor, which can be activated by the CNO IP injection. Then for the inhibition model, we inject the KOR or HN4DI virus into the POH, which can be activated by the SALB or CNO, resulting in the neuron inhibition. So at the same time, we also implant the IPTT or the emitter for the temperature detecting. And after 2 weeks of the recovery, the experiments will begin. So firstly, in the figure A, the injection accuracy was validated by the robust M-cherry and M13 signals. And we also use the PanScan to monitor the neuron activity after the CNO or SARB injection. And as expected, as shown in the figure B and figure C, we can see that the CNO treatment stimulates the ER alpha mPOA neurons, and the SARB treatment inhibit the ER alpha neurons. And then, as shown in the figure D, we find that the selective activation of the ER alpha mPOAH neuron induce a long-lasting inhibition on the cold body temperature and the room temperature. What's more, as shown in the figure E and figure F, the ER alpha activation also reduced the brown edible tissue temperature and the food intake and room temperature. So then we expose the mice to the 6 senses for a cold exposure. And we find that the ER alpha activation still strongly reduce the food intake and the body temperature. So showing a strong effect on inhibiting the cold defense response. And since we have very strong effects on inhibiting the food intake, so next we are wondering if the if this phenotype are secondary to the food intake. So we remove the food and activate the ER beta neuron in the POH and the cold temperature again. The body temperature and the brown edible tissue thermogenesis are still reduced. So the data indicates that the ER alpha can reduce the body temperature and the brown edible tissue thermogenesis. So next, we also investigate the inhibitory effects of the ER alpha neuron in the POH. And as expected, the inhibition of the ER alpha in the POH significantly increased the food intake and the core body temperature and the 30 senses. And these effects are getting more robust and the 37 senses, which indicates the hot exposure. So this data indicates that the ER alpha POH neuron regulate the adaptive response to the temperature challenges. So similar strategies were used in the female ER beta Cree mice to selectively activate or inhibit the ER beta POH neurons. So from the figure B to figure F, we find that the activation of the ER beta also induced the inhibition on the core body temperature, food intake, and brown edible tissue thermogenesis and room temperature. The difference with the ER alpha inhibition is that the ER beta activation cause more short-term effects. And conversely, from the figure G to figure L, we can see that the ER beta POH inhibition increased the body temperature, back thermogenesis, and the food intake and room temperature and thermal neutrality. So this result implies that similar metabolic effects of the ERα and ERβ in the mPOA, consistent with the observation that the majority of the ERβ POH neurons are the ERα positive. And notably, the time scale of the response induced by the ERα or ERβ activation are different. Thus, the ERβ neuron may represent a subpopulation of the ERα neuron, mediating the estrogenic regulation of the energy homeostasis. In contrast, the other non-estrogen, non-ERβ or ERα neurons may contribute to the estrogen-induced topo-like response. Then, to test the chronic metabolic effects of the ERβ inhibition, we generate a mouse model with the ERβ neurons selectively overexpressed and involvely rectifying potassium channel KIR 2.1. Specifically, the KIR 2.1 has been previously used to stabilize the resting membrane potential and decrease the excitability of the hypothalamic neurons. So here, from the figure A to figure D, we can see that the chronic inhibition of the ERβ POH neuron increase the body weight and the adiposity in both sexes. And from the figure E to figure H, we can see if we will switch to the hyperdine, these pro-obesity effects were enhanced by the hyperdine feeding. Notably, we also observed the subtle increase in the energy expenditure and the food intake when the mice were fed with the chow diet and room temperature. And this change was significantly enhanced when the ambient temperature was increased from the 22 Celsius to 30 Celsius. So this data suggests a important role of the ERβ POH neuron in the body weight control and the temperature or nutrition-dependent metabolic adaptation. Thus, our next step is to determine whether the ERα or ERβ expressed by the POH neuron are required for the estrogenic regulation of the energy expenditure or the metabolic adaptation to the temperature or nutritional stresses. So firstly, with stereotaxic inject the AAV-CMV-CRE GFP to the POH of the ERα flox mice to specifically delete the ERα in the POH. From the figure A to figure B, we find that the ERα knockout in the POH did not affect the body weight in both sex and the room temperature. However, it increased the body weight and adiposity induced by the hyperdine challenge, as we can see from the figure C to figure F. And notably, the energy expenditure and the food intake were not affected by the ERα knockout when the mice fed with a chow diet and a room temperature, indicating that the ERα POH neuron is not required for the body temperature control and a mild cold ambient temperature without the nutrition challenge. And then we switch them, we expose this animal to the thermal neutrality. And we find that the ERα knockout mice increase the energy expenditure and the food intake and the rectal temperature. So the data indicates that the temperature-dependent inhibitory effects of the ERα POH neuron on the heat production, food intake, and body temperature. So these metabolic phenotypes are consistent with the observation that the estrogen activates the vPOA neurons through the ERα. So similarity, we inject the AAV-CMV GFP or the AAV-Cre GFP into the MPOA of the ERα beta flux mice to specifically knock out the ERα beta in the POH. And we find that the ERα beta knockout in POH increased the body weight of male and female mice and room temperature while fed with a chow diet, but not on a high-fat diet. And we also observe a significantly decrease in the body temperature, fat mass, and the lean mass in the female ERα beta knockout mice after 12-day cold exposure following the chronic high-fat diet feeding. This data suggests distinct metabolic functions of the ERα and ERα beta in POH during the different nutrition status. So by this chance, I wanted to thanks for everybody's help for this project, and that's it for today's report. Thank you.
All right. Right now it's open for questions. Any questions from the audience? Yeah. I must say, this is a complicated story. I think the first point we're trying to answer is why the single neuron have two receptors expressed, and also those two receptors have opposite functions. One is activating neural activity, another one is inhibiting neural activity. So why they express in the same neuron, and in which condition one receptor is override the other? So that's the question. I think this is a well-known phenomenon, not only restricted to estrogen receptors. Multiple different receptors has this patterns. Yeah, so we're interested about this. Our result basically showing that those two different receptors may have different function during different nutritional status and temperature status. So during specific temperature, probably one override the other. So that's the observations we have. Any questions related to this project?
Sorry, I missed most of it. I had a late night in another meeting this morning, but looks like you're making good progress though.
Thank you. This project has been submitted as an R1 in the past February cycles. Any question, any comments or suggestions are more than welcome. Yeah, we are looking for comments.
Have you already gotten the comments back from the review cycle? Probably not.
Yeah. We do have several important comments, and we're trying to address those.
Okay.
One of the comments is related to specificity of our models, because we are trying to target this neural populations co-express both receptor, but the current technologies we have cannot address this. So we're trying to switch to a different double selection mouse models. Yeah.
Okay. And somebody has that double model?
Nobody have this, but we will generate it.
Oh, okay. You'll generate it. Gotcha.
Yeah,
Okay. Good.
it's a combination of mouse line and also the virus. Yeah. So
Okay.
yeah, we have the alpha Cree and commercially right now is beta Cree is available, and also alpha lipo is available. We can crossing alpha Cree and we can crossing alpha lipo and beta Cree to have this both double selection models, and combining with the virus strain. Specifically is a CRISPR virus strain, we can achieve double selection.
Okay. Were there any kind of more concerns that were harder to address?
I think right now we address most of the concerns for the previous cycles. Right now we drafted as a new submission, so we will see is there any like new comments.
Okay, cool.
Yeah.
All right, well, good luck.
Well, I want to ask you a question.
Uh-huh.
Or kind of global, general sort of thing. Just about, sexual dimorphisms. And, I've taught, based on imprinted genes, I tend to think of them being, related to evolutionarily advantageous adaptations in terms of how we handle energy, and particularly in females, the ability to store energy.
Mm-hmm.
And have reserves, and the ability to store fat is one example of that. And how does it seems to me that regulating body temperature and energy expenditure would be another important component of that. And does that perspective, does your data fit within that kind of big picture of energy, homeostasis, and sexual dimorphisms?
I think so. I think this brain regions is well known as the important brain regions handling both the temperature and the energy homeostasis.
Mm-hmm.
Our hypothesis is in females specifically, this estrogen can act through this neuron populations, express both receptor to regulate both temperature and energy homeostasis. However, during different status, this special neuron populations can switch receptors.
Mm-hmm.
What I mean is those receptors either can internalized or can be degraded so that the other receptor can override the existing receptor to mediate opposite functions. For example, during cold, probably this neuron population can be activated so to generate heat. However, during like a warm temperature, probably the opposite phenomenon will happen. So this is important for female survivals during extreme environmental temperature, and also during this nutritional starvations.
Yeah, so related to that, another important metabolic stress for females is pregnancy.
Yeah, that's correct.
And, postpartum, feeding,
Mm-hmm.
nurturing the young. And, so I wonder whether these nuclei and estrogen signaling under those physiological conditions, which are so important, might be worth exploring. And the role of this, how energy, homeostasis, body temperature, heat generation are kind of being impacted by these, these states, pregnancy being high estrogen, how is the role of this nucleus playing into that? And energy concert, I think putting it in that frame might be interesting and important physiologically.
Yeah, we have some ongoing collaborations with other labs. They focus on prolactin. Yeah, prolactin is super high during lactation phase. So prolactin also related to estrogen. Estrogen is a negative regulated prolactin, and the prolactin also negative regulating estrogen. So they balance each other.
Yeah, do you have any thoughts, just to push it a little further about, the effects of high estrogens? Well, no, well, during pregnancy, energy homeostasis, thermoregulation, the role of this nucleus, the role of those two receptors, in terms of that, metabolic stress state. Do you have any thoughts about that?
I don't have evidence, but all I have is just anticipations. Yeah, yeah.
Uh-huh.
So I would say probably those nutritional status during pregnancy could mimicking hypothalamic feeding in certain
Or starvation.
yeah.
Or starvation, it's
Mhm.
a stage of accelerated,
Mhm, mhm.
lipolysis
Yeah, yeah.
and dependent on fatty acid metabolism, and in
Mhm.
a way, it's kind of you're on the edge of a catabolic state there. So that's why I think it may be very interesting, to think about that.
Yeah, that's true. We don't have the expertise, but we definitely can collaborate with other lab to look into that. Yeah. Yeah, because right now we switch the focus to this metabolic adaptation. Yeah, we more focus on the adaptation phase induced by different stressors.
Well, that's what I'm saying pregnancy is a stressor.
Yeah.
And this system seems like would be particularly relevant, in that kind of stress state that's linked to reproductive functions. So
Yeah.
Yeah, Ping, what Terry's talking about is if you overnight fast a mouse or a human that's pregnant, their ketones go up dramatically compared to a non-pregnant individual. Obviously, the mouse has a little bit of ketones, but in pregnancy, it really goes up too. And so it's a phenomenon that's really not fully understood.
Okay.
yeah, but I think it's to be pregnant and to build another body requires energy.
Yeah,
And
yeah,
it has to come, and it has to come from fat, So that means that the ability to store fat is kind of a precondition for becoming pregnant, like a precondition for being able to successfully hibernate. You got to store fat. So females are good at that, and it's no accident, and this is likely related. But you also have to be able then to mobilize fat efficiently. So switch states, and this from fat storage to tapping into the reservoirs, and and I just think I wonder whether this system here is in the switch, is important, alpha and beta and so on. So it's not what you're looking at right now, but it's evolutionarily as important as the adaptation to cold. That's all I'm saying, right? And maybe more so.
It's an important question.
And likely linked, in terms of overlap, in terms of pathways and physiology. Yeah.
Yeah. Okay. If no further questions, I think we can end the seminar.